Welcome, everyone, and thank you for joining us today for our Hunch and Learn event. I take great pleasure in introducing today's speaker, Dr. Ian Villamagna, my colleague, with his wealth of experience in developing nanoparticle formulations. And today, Ian will be telling us a short story about scaling up nanoparticle formulations, the path from preclinical development to clinical manufacturing. Before we jump in to the talk, I'd like to take a minute to introduce our West team here in North America. Starting, of course, with Ian, today's speaker, field application scientist located in the Bay Area, covering the Northwest Territory. Myself, Viet, his counterpart in the Southwest Territory, who works closely with Kim Killian, the regional sales manager, also in the Southwest Territory. And last but not least, Angel, regional sales director covering the whole West and located in the Bay Area. Before we start, I also would like to mention all our other events happening later this quarter. Webinar by Dr. Gaurav Sahay in April, Journal Club led by myself in May, and another webinar at the end of May presented by Dr. John Lewis. And just a reminder, all of these events are actually on our YouTube link. There's a playlist on the YouTube covering all of these events. If you'd ever want to look back at any of the recordings. So, without further ado, I hand it over to you, Ian.
Thank you, Viet. Yeah, thank you everyone for joining us this afternoon. Hopefully, everyone's having a great day so far, and that we can discuss some pretty relevant topics as you enjoy your lunch if you're here on the West Coast or wherever you are. So the goal for today was to talk about really scaling up nanomedicines and how these constructs go from essentially preclinical development into clinical manufacturing, and to show some of the studies that enable scale up, if you will. So just to start off, as we always start off our talks, we just like to remind people of our vision at Precision Nanosystems, which is to accelerate the creation of transformative medicine that significantly impacts human well-being. And the way that we really do this is we leverage our variety of technology platforms that enable the development of these types of drugs and constructs that we're dealing with today. So this, for example, would be more of our genetic medicine platform, which includes platform technologies in genetic payloads, lipid nanoparticles and lipids for delivery, and a manufacturing platform as well. And we really work to couple those with things like our drug development expertise that we have both in the field, like Viet and myself, and then also back in Vancouver with our team, to really enable our customers to develop these products as quickly as possible. So I think if 2020 showed us anything, it was that we need to be able to take these types of medicines, vaccines or therapeutics really, that are quite complex in nature, and we need to be able to move them through this development process in a really rapid pace to respond to new threats or issues that are occurring. And I think that the RNA vaccines that we've seen right now have been really a great example of that. You know, Moderna, we hear about a lot in terms of how quickly they were able to get their vaccine from conception up to dosing their first patient, and that it was about 45 to 60 days in between that time frame. And so that very rapid time frame, I think, gave us an indication really of how quickly we can get these sorts of drugs to market. And what I would say is that there was a lot of work that was done before this was done to enable that very quick movement, and a lot of de-risking that was done to get to that point. So in terms of scale up of a traditional, vaccine like this example here, or even a therapeutic that we see in this space, typically what you're looking at are 4 to 5 years really of scale up processes and that development of the scale up process. And so this is something that I think, last year we saw that this was an issue that had become exposed quite readily. We needed to have the ability to scale these products up very quickly to meet this urgent need. And what this really requires is a very, I would say, more sophisticated approach to manufacturing, and to doing that at a much earlier stage in the development process. So you're not just trying to model scale up at the end of your process where you have a vaccine candidate or a therapeutic candidate, but you're really looking at this scale up idea at a much earlier stage in development. And so with our technological platforms at Precision Nanosystems, that's really the philosophy behind all of them, is that we are modeling the GMP process at an earlier stage in that development. And what that allows us to do, first and foremost, is reduce the risk in transition from something like research and development up through the scale-up process. So, if we have a really quality product from the start, we can feel more comfortable walking through that scale-up process and modeling some of these studies at an earlier stage in development that will mean something very impactful in a later stage of development. And it also means that we have a better understanding of how these constructs and how these therapeutics will behave and respond down the line when we go to actual clinical manufacturing. So I'd like to talk about some of these ideas about the studies that we look at doing when we look at scaling up these types of constructs and these types of medicines. And I think it's really useful to kind of start by thinking about the past and what we've seen from the past and why this type of idea becomes so critical. And so one really good case study is Doxil. For anyone that's not familiar, though it is pretty ubiquitous overall, I would say, Doxil is a liposomal formulation of doxorubicin, which is a very powerful anti-cancer agent and very useful. It does work quite well for a variety of different areas. The downside being it does come with a large amount of systemic side effects that can really limit our ability to use it in the clinic. What Doxil looked to do was encapsulate doxorubicin within a liposome, which would then basically allow for a decrease in those side effects, those toxic side effects that were associated with doxorubicin, and allow for essentially a more widespread use of this very potent anti-cancer agent. And for all intents and purposes, Doxil worked quite well. And the idea of encapsulating this within a liposome was quite useful. But Doxil itself was actually plagued by really strong and widespread manufacturing issues that really persisted throughout, I would say, even up past 2010. And the reason for that was, again, because these constructs that you're looking at here on the screen, like this example of what Doxil would be, are really quite complex in nature. You're no longer working with one molecule, but you're working with hundreds of thousands of molecules. You're working with lipids and small molecules and stabilizers, and they are more complex. A traditional liposomal production, like something that would have been done for the production of Doxil at a large scale, has a number of steps in it. And so typically, if you were to look at more of a laboratory type scale that we might be more familiar with from an R&D standpoint, it would start with essentially the addition of your lipids and your drugs into solvents, going through a stage of rotary evaporation to form a thin film, essentially, and then rehydrating that film to form these multilamellar vesicles, which you would then go through and impart energy to break down into smaller and smaller pieces, and then go through a purification process to have your final liposomal product. All of this together, depending on how many times you have to go through that downsizing or purification step, can be a number of different steps, upwards of 10, really. What we look to do with our manufacturing platform at Precision Nanosystems is really simplify that manufacturing process from the very beginning. So with the nanoassembler, for instance, you can form these exact same liposomes really in two main steps. So the addition of your components in solvent streams, the mixing of them together happens in one step through the actual manufacturing platform, and then going right into your purification process, where you're able to make these unilamellar vesicles from the beginning. So what that allows you to do is really remove a number of steps from the process. And with each step you're removing, what you're removing along with it is the inherent variability that comes with each step in that development process. And so whereas that 10-step process that occurred with liposomal production in the past was quite challenging. What we've seen with microfluidics and with our platform is that essentially you're able to put your pieces together, formulate through this system, and you do form, as you see on the right here, these unilamellar liposomes very readily. And so I think, we can appreciate right now, whether it's liposomes or lipid nanoparticles or polymeric nanoparticles, a more simplified manufacturing process is probably going to lend itself to not running into the same issues that Doxil ran into with their manufacturing processes. And having just a more simple approach to the manufacturing that delivers a more quality product from the very beginning, the very early stage of development, can be quite beneficial overall. So what I'd like to do now is kind of talk about some of those studies that I mentioned off the top. So, as we're going through the R&D phase and we're thinking about scale up, how do we actually get there, and what do we do to actually determine that we're prepared and ready to go there? And so in order to kind of talk about this, I'll point to a publication from 2020 that came from Yvonne Perry's lab at the University of Strathclyde. And so this lab is a great resource for anyone that's looking or working in this field that would like to look at some literature. This specific paper is done on liposomes, which I think can be quite useful because liposomes are, at least in my opinion, more challenging to work with than something like a lipid nanoparticle. So there's a lot that we can learn from liposomes and apply to other areas. But also, I think they have a great wealth of information coming out of this lab in terms of the studies they've looked at and the type of data they've gotten that we can apply to a number of different places. So here in this publication, what they were looking at was the really rapid scale up and production of these pegylated liposomes. So first and foremost, looking at their production through the microfluidic architecture, downstream processing like solvent removal and the effect on that, looking at loading, essentially, of drug in this case. So for liposomes, they were looking at active loading. And then looking at the final product and how that actually worked through a scale-up feature. So going from that laboratory scale, the instrument and scale production up to a much larger scale of production. And so I'll kind of walk through some of the key studies that are outlined in this paper. As with a lot of the papers, I would say, that come out of this lab, there's a lot of data in this paper, and it's all really good. So it's hard to kind of break down some of these studies and pick out one specific one that I think would be useful. But I think the key here that this paper really explains and goes into well is really when it comes down to talking about scale up, what it really shows is that scaling up is really about modeling that scale up at a much smaller scale and an earlier stage in the development. So what you're looking at here are a collection of the studies that were done at the very beginning for these constructs, where the authors looked at really a variety of different areas with this specific formulation. So what you're looking at here in the top and bottom rows are two different actual formulations. So using different lipids, specifically helper lipids in this case, HSPC and DSPC. And with those two formulations, then in columns, if you will, across this figure, what they were looking at were differences in the flow rate ratio. So how much of aqueous to organic phase they were mixing to form these particles or liposomes. Looking at the total flow rate in column B, so how quickly they're flowing through that microfluidic architecture. And then also looking at different lipid concentrations as well to look at what effect that has on the overall particles in terms of size and dispersity of the population here. And so these studies, I would say, are very commonly done at early stages in development. And what they allow you to gather and gain and learn is essentially how your specific product responds to those manufacturing changes and those changes in the way that you're manufacturing them. I think that there's a lot of good data out there looking at concentration and what effect that has on something like the size and polydispersity. And then even further than that, the efficacy and the overall stability of the particles. And I think that we're getting a lot of data now looking at stuff like the flow rate ratio. and the total flow rate. And so that'll give you an indication of how these particles or liposomes form at specific flow rates, and if you'll be able to tune the size by essentially adjusting those parameters. One of the other things that these authors did at really an earlier stage in development, I think, than what we are sometimes used to seeing, is they were modeling really, I would say, more of the overall efficacy of these particles at a much earlier stage. So looking at essentially the different particles they were able to form using lipid composition, changing the flow rate or the flow rate ratio, what they were able to look at was also essentially how efficiently these liposomes released the doxorubicin that they were encapsulating. So this is something that is a little bit more specific to a liposomal preparation rather than something like a lipid nanoparticle, where with a liposomal preparation, you really need that small molecule that's in there to be able to be released efficiently and readily. And what they were able to show here is that essentially changing the lipid composition had a larger effect on doxorubicin release, whereas changing something like the flow rate ratio or even the loading temperature that they were adding into these liposomes that did not really have a large effect on the release of doxorubicin or the expected efficacy, I would say. These are in vitro studies, so they aren't showing any in vivo data, but I think that they're trying to gauge what that response would be in vivo. One important thing here that I would say for the box at the bottom in D is that one of the things you'll see these authors do all the time in their publications, and I think is really key, is that everything is related back to essentially the polydispersity, the size, and the encapsulation efficiency on these particles that they made, or liposomes in this case. And what we refer to that as typically is the total particle quality analysis. And it's really important to have a good measure of those key attributes for how your liposomal population or your particle population are responding to these changes. These values, while fairly easy to obtain through dynamic light scattering, they're really telling in terms of what's going on with your particle population overall, in terms of what affects something like changing the ratios, changing the flow rates, the lipid composition, what effect is that having on size and polydispersity or encapsulation efficiency? And we can really learn a lot from that measure. We can understand, if particles are aggregating, if we expect for them to release the same amount of drug. We can learn quite a bit from those sorts of measures. Here, what the authors looked at was two different types of release media. So I mentioned that they were doing in vitro studies. And one of the things that I did here on the right, which you see, is they took their PBS release medium and they added essentially a human serum to it. So adding proteins to that mixture. And I would say overall, I think this is kind of what we expect to see. And this is what I think I've seen a lot of in my experience. When you add protein to a mixture, you typically see a little bit of a lower release rather than compared to when you don't have protein in it. But I think this is a really useful study to try and look at essentially, how close can we get to modeling how this is going to be released in vivo in a laboratory setting. And so I think the addition of protein in the way of serum here was quite useful for that. And then finally, here in C at the bottom, what they're looking at is beginning to look at that downstream process and how downstream processing of these liposomes affects them in terms of, again, the size and the polydispersity, and in this case, also the zeta potential. So any formulation that you run, you will have to run through downstream processing in some instance for the most part. And here what they're doing is they're getting an early indication using dialysis of what that process looks like and how it affects these particles. We have seen in the past that some types of particles or liposomes behave and respond much more effectively or differently to downstream processing than others, partially due to things like stability. But with some particle populations, we see a lot of aggregation and a lot of essentially breaking of the particles, whereas we don't see that with some other constructs. And so I think it's a really key study to really do as much research and development on that downstream process at a smaller scale so that you know what it looks like when you get to that larger scale. So you're not dealing with hundreds of milliliters of formulation and then looking at downstream to try and ascertain how that downstream is affecting things. You can model these types of studies at a smaller scale and learn a lot about them. And then finally, towards the end of this publication, which again, there's a lot of data in here, and I would recommend it to really anyone. I think it's a great overall publication and overview of these processes. Towards the end, what they begin to look at is essentially using a different instrument. So using a larger scale instrument to make these same constructs. And so in between mixing these fluid streams together at 12 milliliters at a benchtop or laboratory type scale, and then looking at a larger scale instrument, like you see on the right here, what they're able to see is that across the board, once you get to that larger scale instrument, whether you're flowing at 12 mils per minute, 60 mils per minute, or 90 mils per minute, in terms of size and PDI, you have the same attributes of those particles. This is also true for zeta potential and for the encapsulation efficiency as well, where they didn't see any changes overall, even from switching different instruments either. And so I think what this really goes to show is that by using a more sophisticated manufacturing platform and a manufacturing technology, what you're able to do is really readily and rapidly transfer your processes from that laboratory bench scale into a scale-up method. And really work with a sense of control, knowing that anything you're doing on that bench scale can be scaled up effectively into larger volumes when the time comes. One of the things I like here about this publication as well that you see in D is that they did take the time to do these, I believe, cryo-TEM of these liposomal populations that they made, which can be a really challenging thing to do and very a big time constraint. And so you don't see TEM all that often, but what you're able to see from this is that both at the bench scale and through a scale-up model as high as 90 mils per minute, they are making unilamellar liposomes that look quite similar across the board. And so a really quality product. So some of the author's conclusions here were a little bit far-reaching, and I think that's good overall, because I think that this publication really does show, this type of scale-up method, what potential it has. And so essentially what this showed, or what the authors believe this is going to lead to, is that this effective type of scale-up, where we can take these small-scale formulations and know that they're able to be scaled up very rapidly, will lead us to an era, if you will, of this personalized medicine or just-in-time medicine, where we can essentially, we know what we'll have to formulate ahead of time. We've done a lot of de-risking at a smaller scale, and we'll always be ready to go to move into this more large-scale production when the time becomes required. And so they showed that here with a number of different drugs that they're able to load in these liposomes in a very quick fashion. And they attributed the ability to do this to their research and development studies in an earlier stage, and those studies that we talked about previously. I think this is really relevant right now as well. Obviously, we've seen this for lipid nanoparticles. The ability to take, if you will, a lipid nanoparticle that's preformed or that we know the composition of, and being able to plug and play, for instance, different RNAs within it, is really crucial. We've seen that, and it's going to become more crucial in responding to these types of events that we're going through in very rapid fashion. And so we focused on liposomes here for the start, and I mentioned that I think there's a lot of crossover between something like a liposomal formulation and a lipid nanoparticle formulation. And so I'd like to kind of show that here with some data that we developed at Precision NanoSystems within the past couple of years, where we were looking at the scale up of RNA lipid nanoparticles. So in this study, what we were looking at doing was using an mRNA that encodes for human EPO, or the EPO gene, and looking at it as a way to essentially take the place of EPO therapy, which can be used in anemia patients and is quite commonly used. The benefit for using an RNA that encodes EPO rather than the EPO protein itself is that the current methods of production for that EPO protein, while well studied and well understood, is still quite challenging and does take some significant processes in terms of cell culture. And by using an RNA and a lipid nanoparticle delivery system, you're bypassing a lot of those steps in terms of cell culture, and you have more of a more easy method of development, I would say. So here, what we did is we developed these lipid nanoparticles with that RNA, and we were able to really look through that scale-up process as well, and then finally do some in vivo analysis overall. So in terms of our experimental design, we used 3 different instruments that are part of the NanoAssembler platform, that being the Ignite, the Blaze, and our GMP system as well. And so here, what's important to note and important to know about the manufacturing platform in general is that the microfluidic architecture you're using is the same at the Ignite and the Blaze and the GMP level. And so what that allows us to do is work, again, with the knowledge that anything we're doing at the Ignite level can be readily and rapidly scaled up to go into the Blaze and to then the GMP system. So here we were using Genvoy, which is a lipid mix that we have available to our customers for research use only. We were essentially combining Genvoy and ethanol with essentially 175 micrograms of the RNA in a formulation buffer, and then making different mixing volumes at different rates. So at the Ignite level, 4 milliliters at 12 mils per minute. Then onto the Blaze, we made up to 55 milliliters at up to 90 milliliters per minute. And then into the GMP system, making 325 mils up at 200 mils per minute. These volumes are not the max amount you could make, but it's what we did just for the modeling of this study. And so across, yes, yeah.
Sorry, before we move on, I think there's a question that's quite relevant to that slide about why can we run 90 mils per minute on the Blaze, but not on the bench scale and on the Ignite?
Sure. Yeah, no, it's a great question. So our microfluidic architecture, what we call next-gen microfluidics, it does have a different architecture than a lot of other microfluidics do. And within that architecture, we do not have any 3D structures that actually develop or actually allow for the mixing to happen. So because of that, what we're able to do is scale that architecture geometrically and still preserve the physics of mixing of those fluid streams. But as we've scaled geometrically, we then have the ability to move at faster flow rates. So we're able to move at faster flow rates, still mix those fluids in the same fashion, but essentially just doing it at a much larger scale overall. And so the microfluidics does scale with the instruments as well, not to a point where we'd be able to see it. I mean, these are still extremely small, but it does scale along with the actual instrument platform. So as I mentioned, and as I think we saw with that publication that we talked about, here we went back to looking at total particle quality analysis to determine what we had made across the board and across this platform. And so in terms of hydrodynamic diameter or the size, and then the polydispersity or how monodispersed the population of particles was, what we were able to see is that there was no difference across the board, significant difference at least, when using the Ignite versus the Blaze versus the GMP system. So we are able to make these particles at the 12 mils per minute in the lab scale, and then also at even as far as the clinical manufacturing scale, 200 mils per minute on the GMP system. Furthermore, encapsulation efficiency was very consistent across the board as well. with all of these constructs having just about above just over 90% encapsulation efficiency of that RNA. And then so very telling, I think, in terms of in vivo data here, again, looking across the board, whether these were made at the lab scale with the Ignite or on the Blaze, which is, late preclinical stage, if you will, or at the GMP stage for clinical manufacturing, we get the same essentially percentage of both hematocrit and the same readings for the serum EPO levels. So from an in vivo efficacy standpoint, in this mouse model, what we were able to show is that particles, regardless of where they're made in the platform, do have the same biological effect. And then also on the right of these graphs, the effect of tangential flow filtration is not something that we see as compared to, for instance, an Amicon filtration. So again, looking at the scale up, we then also modeled the downstream processing for that scale up, and we still don't see that a difference from those types of processes. And so just to go through some conclusions here today, I think we talked a lot about essentially the scale-up process and kind of moving through that, some of those earlier stage studies that can really help with the scale-up process. Again, the way that we really try to enable our customers to move through this process is through our technology platforms. And so working with our customers to essentially walk through those steps to de-risk the movement through the development process. And so obviously today, we mentioned a lot about the NanoAssembler platform, the manufacturing platform, which as it stands looks like this. So comprised of 4 instruments, all the way down from the Spark, which is meant for very low volumes and really as a rapid screening tool, up into the Ignite, the Blaze, and the GMP system, which are really meant to walk you through that development process with your own therapeutics or vaccines that you're working on. Important to note here, again, the microfluidic architecture is retained across this platform. So the same architecture that you use on the Ignite is present on the Blaze and the Spark as well, actually, all across the board. So anything you do at any level, we feel fairly comfortable saying can be transferred and walked through this scale-up process. And then finally, just to close off, I think we get this question a lot about, what does this pathway to manufacturing really look like, and what abilities are there to essentially go through this manufacturing pathway? This is an example of one of our customers that we're working with in our region. And this is the path that they were able to take to essentially get to clinical manufacturing. And so it started in October of 2018. It actually predates my time here with Precision NanoSystems, where they were able to begin work at that laboratory bench scale on an Ignite, and then move through essentially the preclinical process development. So it took about a year for them to get through that bench scale to do a lot of these studies we talked about today and really model their overall formulation. Moved into preclinical process development with the Blaze in January of 2020. And we're able to run really a number of studies even in 2020 with everything that we know that happened and the shutdowns that occurred, and really run a number of studies to look at essentially that scale-up process and how their formulation responded to faster flow rates and larger batches and downstream processing. As of August 2020, they had taken possession of one of the GMP systems and went through some engineering runs. And in March of this year, after all the legal was worked out, they were able to transfer their method to a CDMO and prepare for clinical manufacture of their product. So all in all, a fairly accelerated timeline here for this path to scale up. And so with that, I see that I've seen, as I've been talking here, the number pop up. So happy to take any questions at this point. If you're interested in learning more, you'd like to reach out and chat with us, whether you're a prospective customer or someone that we already talk to all the time. As always, feel free to reach out to really any one of us. We're happy to point you to the right team if you're not sure what region you lie in. Always happy to talk about any of your development and what you think you may be able to do or how we could assist in enabling you to drive your products through to manufacturing.
Thank you for the talk, Ian. Really, really exciting.
And so I think if that's all we have, then for today we'll go ahead and close it off. Just again, thanks everyone for joining. Really appreciate it, and feel free to reach out at any point. And thank you very much for all the questions. It was fantastic. We look forward to working with you in the future.